this video we are going to learn about some disconnected topics about molecular biology like ribosome structure and function and split genes along with chloroplast genome. We will be learning about all this. So tRNA is here, amino acids large subunit A site, P site, mRNA is produced. This is a picture which represents ribosome activity. Synopsis. Introduction. Ribosomes in prokaryotes and eukaryotes. Ribosome structure. Functional sites of ribosomes. Models of ribosome structure. Composition of ribosomes and reconstitution experiments. So let's have an introduction about this topic. What are ribosomes? A ribosome is a complex molecular machine found inside the living cells that produce proteins from amino acids during the process called protein synthesis or translation. The process of protein synthesis is a primary function which is performed by all living cells. Ribosomes are specialized cell organelles and found in both prokaryotic and eukaryotic cells. Every living cell requires ribosomes for the production of proteins. A ribosome is a complex of RNA and protein and is therefore known as a ribonucleoprotein. It is composed of two subunits, smaller and larger. Discovery. Ribosomes were first observed in mid-1950s by Romanian-American cell biologist George Emil Palade using the electron microscope as dense particles as granules. Then Albert Claude Clistin de Dieu and George Emil Palade were jointly awarded the Nobel Prize in Physiology or Medicine in 1974 for the discovery of the ribosome. Nobel Prize in Physiology or Medicine 1974 for the discoveries concerning the structural and functional organization of the cell. Ribosomes in prokaryotic and eukaryotic cells. Prokaryotes. Ribosomes often occur freely in the cytoplasm. Ribosomes often occur freely in the cytoplasm. Usually 70S ribosomes are seen with two subunits. Then eukaryotes. In eukaryote cells, there are ribosomes occur either in the cytoplasm or remain attached to the outer surface of the membrane of endoplasmic reticulum. In eukaryotic cell, 80S ribosomes are seen. Then two types of ribosomes based on the sedimentation coefficient S. Two types of ribosomes have been recognized. 70S ribosome, compartmental smaller in size and sedimentation coefficient is 70S. 80S ribosome have sedimentation coefficient 80S. Swedberg unit. Then structure of ribosomes, then grand large peptide particle. Each ribosome is porous, hydrated and composed of two subunits. One ribosomal subunit is larger in size and has a dome-like shape, while the other ribosome subunit is smaller in size and occurring above the larger subunit and forming a cup-like structure. In prokaryotes, 70S ribosome exists in two subunits, 50S and 30S, but in eukaryotes, 80S ribosome also consists of two subunits, 60S and 40S. 50S ribosomal subunit is larger in size and is the size of 160A to 180A. The 60S ribosomal unit is dome-shaped and larger in size. The 30S ribosomal subunit is smaller in size and occur above the 50S subunit like a cap. The ribosome remain attached with the ER, nucleus, etc. The 60S remain attached with the membrane. Both the subunits remain separated by a narrow cleft. The 40S ribosomal subunit is smaller in size and occur above the 60S subunit, forming a cap-like structure. The two ribosomal subunits remain united with each other due to high concentration of Mg2 plus ions. When the concentration of Mg2 plus ions reduces in the matrix, both ribosomal subunits get separated. Actually, in bacterial cells, the two subunits are found to occur freely in the cytoplasm and they unite only during the process of protein synthesis. At high concentration of Mg2 plus ions in the matrix, the two ribosomes become associated with each other and known as the dimer. Further, during the protein synthesis, many ribosomes are aggregated due to common messenger RNA and form the polyribosome or polysome. Polyribosome or polysome, you could see structure. 70S ribosome and 80S ribosome, prokaryotic ribosome, 50S subunit and 30S subunit, 70S. Eukaryotic ribosome, 60S subunit and 40S subunit, 80S. Functional sites of ribosomes. There are 3 binding sites on each ribosome. They are A site, P site and E site. The A site, the aminoacyl site or A site, binds the incoming aminoacyl tRNA. P site, the peptidyl site, binds the tRNA carrying the growing polypeptide gene. Peptidyl site binds the tRNA carrying the growing polypeptide gene. E site, the exit site, binds the departing uncharged tRNA. The smallest subunit has a binding site for mRNA. The codons on the mRNA determine the type of tRNA binding with A and P sites. mRNA binding site, E or exit site, P peptidyl site, A aminoacyl site. 30S subunit. E site, P site, A site. 50S, 30S mRNA binding site, growing peptide J mRNA. Models of ribosome structure. 2 models have been proposed to explain the structure and mode of association of the subunits of bacterial ribosomes. They are quasi-symmetrical model proposed by Whitman, then the asymmetrical model proposed by Lake. First, quasi-symmetrical model. According to this model, the larger subunit is almost bilaterally symmetrical and the smaller one irregular. Quasi-symmetrical model. According to this model, the larger subunit is almost bilaterally symmetrical and the smaller one irregular. The larger subunit has a spherical base and 3 protuberance. It is comparable to an armchair that has a circular seat. Its spherical base forms the seat for the smaller subunit. The prominent central protuberance forms the supporting back and the lateral ones form the armrest. So the smaller subunit is slightly bent and bipartite rod comparable to a telephone receiver. It has a cleft which divides it into smaller head and large body or base. The smaller subunit is slightly bent and bipartite rod comparable to a telephone receiver. It has a cleft which divides it into a small head and a large body or base. Body has elongated extensions on both the sides. The long axis of the smaller subunit is placed at right angles to the central protuberance of the larger one, and its cleft directly faces the vaulted seat of the larger subunit. As a result of this association, a gap or groove is left in between the 2 subunits for the orientation of mRNA. So dent, head, cleft, tunnel, base, 550S subunit, vaulted seat, lobe, lateral crest or arms, 30S subunit, body, central protuberance, etc. can be seen. That is the Stoffler and Whitman's model of 70S ribosome. Then Lake's asymmetrical model. According to this model, both the subunits are asymmetrical. The larger one has a spherical body or base and 3 major protuberance, one central and the other lateral. One of the lateral protuberance is long and stalk-like and the other one has only a ridge. The smaller subunit has an indentation which divides it into an upper head and a lower body or base. Extending from this base is a small platform. In between the platform and the head is a cleft. It is a site for codon-anticodon interaction and also for the binding of the initiation factor of protein synthesis. The smaller subunit is positioned over the larger one and its head is aligned to the central projection of the larger one. A groove is left in between them for the orientation of mRNA. Central protuberance, head, cleft, platform, base, smaller subunit, valley, ridge, stalk region, then ribosome. Head, platform, base, small subunit plus large subunit, ridge, central protuberance and stalk is equal to ribosome. Composition of ribosome. Ribosomes are made up of ribosomal proteins and ribosomal RNA. Ribosomes are usually made up of 3 or 4 ribosomal RNA molecules anywhere from about 40 to 80 different ribosomal proteins. Each ribosome is composed of 2 subunits, a large one and a smaller one, each of which has a characteristic shape. First, ribosomal RNA. The 70S ribosome contains 3 types of ribosomal RNA, 23S rRNA, 16S rRNA and 5S rRNA. The 23S and 5S rRNA occur in the larger 50S ribosomal subunit, while the 60S rRNA occurs in the smaller 30S ribosomal subunit. Assuming an average molecular weight for one nucleotide to be 330 Dalton, one can calculate the total number of each type of rRNA. Thus, the 23S rRNA consists of 3,300 nucleotides, 16S rRNA contains 1,650 nucleotides, and 5S rRNA include 120 nucleotides in it. The 80S ribosome contains 4 types of rRNA, which are 28S rRNA, 18S rRNA, 5S rRNA, and 5.8S rRNA. The 28S rRNA, 5S and 5.8S rRNA occur in larger 60S ribosomal subunit, while the 18S rRNA occurs in the smaller 40S ribosomal subunit. About 60% of the rRNA is helical and contain paired bases. This double-stranded region are due to hairpin loops between the complementary regions of the linear molecule. The 28S rRNA has the molecular weight 1.6 into 10 raised to 6 Dalton, and its molecule is double-stranded and having nitrogen bases in it. The 18S rRNA has the molecular weight 0.6 into 10 raised to 6 Dalton and consists of 2,100 nucleotides. The 80S and 28S ribosomal rRNA contain a characteristic number of methyl groups, mostly as 2O methyl groups, mostly as 2O methyl ribose. The molecule of 5S rRNA has a clover leaf shape and a length equal to 120 nucleotides. The 5.8S rRNA is intimately associated with the 28S rRNA molecule and has therefore been transferred to as 28S associated ribosomal RNA. Then ribosomal proteins. Each 70S ribosome of E. coli is composed of about 55 ribosomal protein. Out of this 55 proteins, about 21 different molecules have been isolated from the 30S ribosomal subunit and some 32 to 34 proteins from the 50S ribosomal subunit. The primary structure of several of these proteins has been elucidated. Most of the recent knowledge about the structure of the ribosomal protein has been achieved by the dissociation of ribosomal subunits into their components rRNA and protein molecules. Then metallic ions. The most important low molecular weight components of ribosomes are the divalent metallic ions such as Mg2+, Ca2+, and Mn2+. Then reconstitution experiment. When ribosomes are centrifuged in a gradient of 5 mCsCl, about 30 to 40% of the protein is lost. Simultaneously, some other proteins called split proteins are released from the ribosome. When the split proteins are added to their corresponding core particles, the ribosome is reconstituted. Some of the split proteins are appropriately specific for each ribosomal subunit. The split proteins have been further fractioned and divided into acidic and basic protein. Nemura et al. In 1968 fractioned at least 6 different groups of proteins in the ribosome. The reconstitution of functional 30S subunit has been achieved by the addition of 16S rRNA to a mixture of proteins from the 30S subunit. Following a similar procedure, the 50S subunit has also been reconstituted. Along with the subunit, the whole ribosome is also assembled. In vitro reconstitution is a powerful tool for investigating ribosome functions and biogenesis as well as discovering new ribosomal features. Interrupted genes. During 1970, in some mammalian viruses or adenoviruses, next topic, which is not at all related to ribosome structure and function, but still we are going to learn them together. That is the peculiarity of this video lecture. We'll be learning totally unrelated topics together, random topics. During 1970, in some mammalian viruses or adenoviruses, was found that the DNA sequence coding for a polypeptide were continuously but were split into several pieces. Therefore, these genes were variously named as split genes or introns, interrupted genes or intervening sequence, inserts or junk DNA. For the discovery of split genes in adenovirus and higher organisms, Richard J. Roberts and Philip Sharp were awarded Nobel Prize. Split genes were independently discovered by Richard Roberts and Philip A. Sharp in 1977, for which they shared the 1993 Nobel Prize in Physiology or Medicine. The first observations of interrupted split genes, genes in which there are non-coding intron sequences between the coding exon sequences, were made in animal viruses in 1977. Introns are exons. Split genes appear to be complex and fragmented genes alternating coding and non-coding region. The non-coding region are collectively called junk DNA, functionless as DNA. The coding region containing actual information of the genes are interrupted by few to several known coding sequences called introns from intervening sequences, which are spliced out after transcription. Such genes are called split genes since their coding sequences are split into several particles or parts due to the introns. Then interrupted genes, exon, intron, exon like that. Then transcription, primary mRNA, splicing, mature mRNA. Look how this portions of exons are only present in this primary, mature mRNA. Introns are, removed from that. Introns and split genes are very rare or Introns and split genes are very rare or absent in prokaryotic mitochondrial and chloroplast DNA, but relatively numerous in most eukaryotic nuclear genome. All the exons undergo transcription and translation, so they find their expression in the primary interrupted gene and final product, pre-mRNA, mature mRNA, and protein. Introns are only transcribed to the pre-mRNA and sliced out during post-transcriptional chemical processing of immature mRNA. Gene, exon, intron like that. Promoter is also there. So transcription, RNA, splicing, mRNA. Amino acid chain, post-transcriptional modification, later protein. Then mRNA, splicing. In most eukaryotic genes and some prokaryotic ones, the initial RNA, that is transcribed from a gene's DNA template, must be processed before it becomes a mature messenger RNA or mRNA that can direct the synthesis of protein. One of the steps in this process, called RNA splicing, involved the removal or silencing, splicing out of certain sequences referred to as intervening sequences or introns. The final mRNA is consisting of the remaining sequences called exons, which are connected to one another through the splicing process. Nuclear pre-mRNA splicing is catalyzed by the spliceosome, a multi-megadalton ribonucleoprotein complex. Both the conformation and composition of the spliceosome are highly dynamic, affording the splicing machinery its accurately and at the same time flexibility. The mechanism of mRNA synthesis from a DNA containing introns can be explained as RNA polymerase may synthesize both introns, exons, and processing of transcripts occur later on. The transcripts correspond to the introns are removed. The first reports of split genes were provided by exon genes of adenovirus. Hexon gene of adenovirus. The DNA of adenovirus is double-stranded and codes for 20 proteins. 8 out of the total genes are called late genes because they transcribe mRNA late in virus life, and 8 late genes transcribe the singular RNA, which is later cut into 8 mRNAs. The total length of the 8 mRNA however less than the length of the original transcript. When one of these genes was paired with the mRNA, the DNA formed 3 loops which represent the unpaired regions. This indicates that the heron mRNA was transcribed by 4 different regions, unlooped regions, and that the 4 segments were then joined together. The looping is means to exclude unwanted DNA that does not transcribe mRNA or introns. Hexon gene of adenovirus with its transcribed mRNA, A, B, and C represent regions of DNA which do not transcribe RNA. Then evolution of split genes. Before the discovery of split genes in 1977, all the genes are analyzed in detail were of the bacterial type, bacterial genes. Bacteria were considered to resemble with the simpler cell from which the eukaryotes must have been evolved. Now it is supposed that split genes are the ancient condition and bacteria lost their introns only after evolution of most of their proteins. Evidence for the ancient origin of introns has been obtained by the examination of the gene that encode the ubiquitous enzyme triose phosphate isomerase or TPI. TPI is coded by a gene that contains The 6 introns in vertebrates of this are present at the same position as in maize. This shows that fewer introns were present in the gene before evolution of eukaryotes, also 9 years ago. The TPI plays a key role in cell metabolism that catalyzes the interconversion of glyceraldehyde 3-phosphate and dihydroxyacetone phosphate, a central step in glycolysis and glycogenesis. By comparing the enzyme in various organisms, it appears that the TPI evolved before the divergence of prokaryotes and eukaryotes from a common ancestor cell, progenote. Progenote, intron, exon, E. coli, Saccharomyces cerevisiae, Aspergillus, maize, vertebrates. That billion years ago evolutionary timeline is also shown in this picture. The unicellular organisms under a strong selection pressure minimizes the superfluous genome in their cell, whereas there is no such pressure on multicellular organism. That is why Aspergillus has 5 introns and Saccharomyces has none. Precise loss of introns would have occurred by deleterious reaction in prokaryotes. The loss of introns require the exact rejoining of DNA coding sequence. Outline of evolution of particular gene. The most likely source of the information is needed for such event in an mRNA transcript of the original gene from which introns are to be removed. The mRNA may be copied back into DNA by reverse transcriptase. Features of split genes. The organization of split genes would be consistently the same in all tissues of an organism. Introns all by themselves don't have any coding function, but exon and intron combination code for mecharase. Mecharase are the protein encoded by exon and intron combination. They catalyze the excision of introns from the primary transcript of introns from the primary transcript and subsequent splicing exons. Exons have precisely the same order in the split gene and also in its primary and secondary mRNA transcripts. The presence of introns is neither essential nor deleterious to the organisms. All the split genes are begin with exons and end with exon. Primary mRNA transcribed from split gene is known as heterogeneous nuclear RNA, hnRNA, or precursor mRNA. It might be the exact replica of split genes with alternating exons and introns. Next topic, chloroplast genome. What is genome? A genome is the complete set of genetic information in an organism. It provides all of the information the organism requires to function. The study of the genome is called genomics. Originally genome. Originals are the energy factors of eukaryotic cells. Mitochondria found in both plants and animals. Chloroplast found in plants. Chloroplast. Chloroplasts are organelles that conduct photosynthesis. The photosynthetic pigment chlorophyll captures the energy from sunlight, converts it and stores it in the energy storage molecules. The most important function of the chloroplast is to synthesize food by the process of photosynthesis. Chloroplast visible in the cells of Bryum capillary, a type of mouse. Chloroplasts. Plant cell chloroplast structure: outer membrane, inner membrane, stroma, lamellae, stroma, then granum, stack of thylakoids, intermembrane space and thylakoid. Then chloroplast genome. The chloroplast genome is a circular DNA containing some number of genes necessary for functioning of the chloroplast and maintaining their structure. The size of the chloroplast DNA varies within 120 to 180 TBP. Located on each chloroplast contains around 100 copies of its DNA in young leaves, declining to 15 to 20 copies in older leaves. Size of mitochondrial and chloroplast genome. If you need to read this, just pause the video and read. Thank you. Chloroplast DNA is the DNA present in the organelle chloroplast. It is sometimes called chloroplastosome. The presence of DNA in chloroplast was first suggested during the early 1950s. cpDNA is typically circular and consists of base pairs ranging from 12,000 to 1,70,000 long. It has about 120 genes. The chloroplast has at least 3 membrane systems: outer membrane, inner membrane, and thylakoid system, the site of photosynthesis. The stroma, which is the matrix of the chloroplast in between the grana, contains cpDNA, enzymes, molecules, and ions. Then biological reactions. cpDNA replication is hypothesized to occur by a double displacement loop, D-loop mechanism. D-loop replication is a proposed process by which circular DNA like chloroplasts and mitochondria replicate their genetic material. In many organisms, one strand of DNA in the plasmid comprises heavier nucleotides, relatively more purines, adenine and guanine. This strand is called the H, heavy strand. The L, light strand, comprises lighter nucleotides, pyrimidines, thymine and cytosine. Replication begins with replication of the heavy strand starting at the D-loop, also known as the central region. A D-loop is a short portion in circular DNA that has 3 strands instead of 2. The middle strand, which is complementary to the light strand, displaces the heavy strand and forms a displacement loop or D-loop. Each D-loop contains an origin of replication for the heavy strand. Full circular DNA replication is initiated at that origin and replicates in only one direction. The middle strand in the D-loop can be removed and new one will be synthesized, and it is not terminated until the heavy strand is fully replicated, or the middle strand can serve as a primer for the heavy strand replication. As the heavy strand replication reaches the origin of replication for the light strand, a new light strand will be synthesized in the opposite direction as the heavy strand. The D-loop region is important for phylogeographic studies. Chloroplast DNA, D-loop. Biological functions. Chloroplast DNA encodes for proteins and RNAs essential to proper metabolism. In particular, it encodes rRNA, tRNA, ribosome proteins, 4 RNA polymerase subunits. Rubisco is the major protein component of chloroplast stroma. Apart from protein synthesis and photosynthesis, cpDNA has also a role in synthesis of 11 subunits of a protein complex that mediate redox reaction to recycle electrons. It shows applications in genetic engineering.